Check. Hello, mic check.
Okay, good afternoon, everyone. So it's my great pleasure to introduce today's Colloquium speaker, Dr. Mukund Tatai from NCBS, Bangalore. So Mukund did his PhD from Cornell University, okay, undergrad from Cornell University in physics, and he calls him as a physicist by training, an accidental biologist, right? And he did his PhD from MIT in physics. And since then, he joined NCBS and he is there in NCBS since then. He is a recipient of Infosys Award for the last year in physical science category. And his research, he works on a very interesting topic of how things move, if I have to say in a very simple way, how things move inside the cell in a very complex, cellular environment, and then some aspect of membrane trafficking as well. So with this introduction, I invite Mukund here, and we're looking forward to,
Hello? Great. I hope you can hear me way in the back. Thank you very much for the introduction, and it's an absolute honor to be giving this colloquium. And this is the second time I've spoken about this very recently. It's all very recent work, but a lot of what I'll talk about is not the recent work, but the overall area of science in which I'm working. And I think I've really projected this to the students in the audience to show you how exciting this field actually is. And just to motivate it, the kind of thing that you rarely get to think about if you're doing biology are these counterfactuals. What could have happened if things were different? But astrobiology has become a real field now, and there are people who spend all their time thinking, what would life look like if it was on other planets? And this is interesting because the only example of life we have is here. So the question is how you actually take what we know about how life works and then extrapolate from one example about how things work elsewhere. You would think this is impossible, and maybe you're right, but people have made an attempt at it. And if I were to distill some of the thinking from that kind of very, very ambitious question, this triangle here is something that very quickly you focus down upon. So one thing we've known for a long time is that Darwinian evolution seems to be the only game in town when it comes to, as Richard Dawkins put it, this sort of crane to lift up complexity. Equilibrium systems certainly don't do it. Even out of equilibrium systems generate patterns which may look complex, but then if you actually poke around and see how functionally rich they are, they're not the same as living systems. They're not the same. So Darwinian evolution is a way for standard physical and chemical processes to nevertheless appear to be directed towards very, very complex functional achievements. The other piece, which is essential for Darwinian evolution, is the notion of information or heredity. The parents look like their offspring, but slightly different. And this we know now is because there's sort of genes, there's a genome that contains the program or the information needed to make the organism. So it's very hard to imagine something like Darwinian evolution happening without an information storage mechanism. It doesn't have to be DNA, but it has to be something. It's basically a low noise place for information. Pre-life evolution was non-Darwinian. It didn't necessarily have information. Once you got this information storage medium, DNA and RNA, evolution really accelerated. So it's reasonable to speculate that on other planets, information is at the heart of living things. Yes?
Sorry.
Yeah.
But what exactly, in 2 sentences, is Darwinian?
Darwinian evolution has three very basic facts. So one is that offspring generally look like their parents. Okay, and this is very interesting because the information actually goes through a very, very small cell. And so the question of the biology of how that works is fascinating. Darwin didn't care about that. The second piece of that is that children don't look exactly like their parents. And so there is a natural generation of variation in a population. I'll take 4 pieces. The third is that there are constrained resources. And so you cannot sustain an infinite population. And so there's going to be some culling. Now, if you put these 3 pieces together, then it will seem obvious then that of the limited set of variation that you're allowed to have in the next generation, those that will survive are the ones that are able to more efficiently propagate themselves. That's Darwinian natural selection. Yeah. Now, the piece of this that's not obvious is the question of cells. Now, on Earth, all life on Earth today is made of cells. But does this have to be true for life on other planets? Now, what is a cell? You could also ask what's the definition of a cell. A cell is a bounded thing. There's a notion of the outside world or the whole universe and the inside. And the reason cells, at least on Earth, seem to be very efficient agents of Darwinian evolution is if a cell comes up with an innovation, then it can retain the fruits of that innovation for itself, right? It's a private good. It can be very selfish. If you actually share your innovation with everybody, then it tends to be that in this kind of game, you don't actually survive, that people take advantage of you. So the idea of a boundary within which the innovations of the information content are shared, which is a cell. It doesn't matter that the cell is this scale or membrane bonded, but the idea of a boundary is very important. Okay, so this is what actually I've always been interested in biology. I started in physics. I was doing condensed matter physics, and I was looking at how complex systems interact and so on. But in biology, the cell seems to be a wonderful scale at which to start thinking of things, because as a physicist, you can ask, mechanically and physically how things work inside the cell. You can also ask how groups of cells work together, but somehow everything focuses down onto that one unit. It's the most important unit in biology from which all complexity follows and the understanding of which is absolutely central. Okay, please stop me with any questions you might have. So the story I'm going to tell you today is the story of life on Earth and how the cells that we know on Earth came about over historical time, over billions and billions of years. Okay, so there's a few lessons that so over centuries, biologists, naturalists have put together about how life works, and each of them is very surprising. This was very surprising at the time that it first came about, and I'll tell you who came up with this, but this is a fact, which is falsifiable, and it hasn't been falsified. All life, whether it's microscopic, amoeba or bacteria, whatever it is, or macroscopic, blue whales or human beings, or trees, is made of cells. It's not obvious, didn't have to be this way, but it is this way. So how do we know this? So 1600s, this actually had to wait for the advent of a certain type of technology, which is good microscopes. So both Van Leeuwenhoek and Robert Hooke made microscopes, and they were curious about their living environments and their non-living environments, but really surprising that they were contemporaries, and they gave us 2 orthogonal aspects of life. So when Leeuwenhoek was interested in small cells that were free living, I mean, he didn't call them cells, but these animalcules, which any sort of dirty ditch water you look at is teeming with living things. And he had a very fine hand, and he recorded every cell that he saw through this microscope, which is holding in his hand. These were, very complicated cells like these ciliates, but he also actually made drawings of bacteria as he saw them with the naked eye through the microscope. And so these seem to be little animals. They had intent. They went around. They were predators, right? So any ecosystem feature that you could imagine, in a forest, for example, also took place in a little droplet. At the same time, during the same years, Robert Hooke was interested in a macroscopic organism, plants. And when he dissected these organisms and looked at their microstructure, he found that they were broken up into these little compartments here. And he called them cells because they reminded him of the cells of a monastery, for example. Okay, so the notion of a boundary. So, and this is it. There's no other way that living systems work. Whether you're a big tree or a whale or a human or an individual cell, everything is made up of cells. It's pretty astonishing that these cells are basically just groups of these kinds of cells. Okay, fine. Now, after that, and as the cell theory developed, it sort of became it seemed to be obvious that there were 2 types of cells, and it's those 2 types that I mentioned, those that live in groups and those that do not. And this was sort of seen as a natural divide, a very basic definitional divide in biology. Yes? Viruses don't come here at all. They're not cells, and they need cells to replicate. And there are many things that are virus-like also, little bits of DNA that replicate within a genome. There's a whole spectrum of those, and those are very clearly distinguishable from cells. Okay, they need cells to propagate. They're not living. No. They are processes. They're sort of emergent processes of life, but very much like DNA replication is an emergent process. But they are not themselves living. They need a whole system around them to survive. Similarly, yeah. Good. So the divide, it turns out, is not unicellular, multicellular, but it's this funny words, okay, prokaryote, eukaryote. Now, I know all of you know these words, but it is surprising how recently these words actually entered the textbooks. It was literally in the 1970s. This is an ongoing story, and the reason we know about this, again, is due to an advent in technology. It's due to electron microscopes. So when people started to really look inside the cells and see how they were made up microscopically, then it turned out that indeed there were sort of two types of cells, and they did not correlate completely with this unicellular, multicellular. Okay, so let me show you what I mean. So in the 1960s, after doing this so-called ultrastructure, electron microscopy, people looked inside cells and again tried to classify, because it's the first thing you do when you approach a new biological system. There were many different kinds of cells, but they seemed to fall into a few brackets. And people realized that something they thought they really had a handle on, which is bacteria. They were causing infections. They already had antibacterial agents. I mean, this is the 1960s, and there still was no definition of a bacterium. Okay, a formal biological definition. And so this very, very famous paper by Stanier and Van Niel articulates the problem. Since the earliest days of microbiology, hundreds of years, okay, the biological nature and relationships of bacteria have been subjects of perennial discussion. Why have these questions obsessed some members of each succeeding generation of microbiologists? There can be no doubt about the principal reason. Any good biologist finds it intellectually distressing to devote his life to the study of a group that cannot be readily and satisfactorily defined. If you can't define it, you can't study it, okay? And they came up with a definition, which was, in retrospect, a very weird way to define it. Stanier and Van Niel could do little more than define the prokaryotes, sorry, those are bacteria, in negative terms in relation to the eukaryotes, which are like the amoeba and the cells that make plants and animals. Eukaryotes had a membrane-bound nucleus, a cytoskeleton, intricate system, internal membranes, mitochondria, blah, 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 blah. Prokaryotes were smaller and lacked all these structures. They lacked okay? So, eukaryotes were defined first, and prokaryotes were the anti-eukaryote because they lacked all these structures. And this is what became evident after the advent of electron microscopy. Now, this was the 60s, and really a very rapid time in sort of eukaryote origins and origins of cells work, because very soon after this, even this dichotomy was broken. It was broken by this man, Carl Woese. So Carl Woese, genius, he did sort of the first large-scale sequencing experiment. Sequencing means you go out, you get a sample, and you use methods of so-called DNA sequencing where you can read the bases, A, T, G, C, and you can literally read the code on a nucleic acid strand. And so what was realized was that, that there's a certain type of gene that's common to all living things. And so if he was able to amplify that fragment and sequence it, and then use just math or statistics to look at the distance between the sequences instead of worrying about the distance between the morphologies, right? Then he could actually convert this classification problem, which is very subjective, into a pure statistical analysis problem, phylogenetics, which is very quantitative. And this is the way to classify living things. Yeah, and of course they should correlate. And so what he did was he sequenced, many samples, and he just ran it through his mathematical analysis and made a tree. And let me explain what a tree is. A tree, Darwin's idea of descent with modification. Everything in biology is related by trees. You have parents, you have offsprings that are diverging from their parents over time. That's a time-dependent tree. This is a distance-dependent tree. Two branches, the distance between them is the walking length across the tree. Okay, and so you can make a tree to explain the distance of all the pairwise distances of all the sequences of the roots. And what Woese discovered is it's not a two-bundle tree, which would be prokaryote, eukaryote, but actually three. Okay, it was bacteria, which we knew about, and then a formerly cryptic group of unicellular eukaryotes, which are called archaea, also very small without any internal architecture. And then you have the eukarya, which is us and the amoeba. So I'm really showing his picture here and this because this classification of life has not been appended since Carl Woeser's time. He was absolutely right. And we've not found any groups of life on Earth other than the bacteria, the archaea, and the eukaryotes. So far, there are no others. So the next question then becomes, how do these three things relate to each other? Did we come? Yeah.
Very sorry. So how are the archaea different, just in terms
Oh,
of...
they're very different. They use totally different kind of ribosomes. They use totally different kind of lipids. They are so different molecularly. I mean, they look the same. Okay, they live on your teeth. They're not very exotic. But they are so different that there are some theories that they cellularized independently. They are so different at the microscale. Yeah, so you can't put them together and make a cell. Okay, but hold that thought. Okay, so looking at this tree, there's no time axis. So what you have to do, as soon as you look at it, you say, where's the root of the tree? You pull up the root and you let time flow from the bottom. So is the root here, that you had something leading to eukaryotes and then going to bacteria and splitting over there, right? And then you can see this fine print over there which says the root is over here. And that would seem to say that there's some common ancestor. On the one side, you get bacteria. On the other side, it then splits to make archaea and eukaryotes. And so that was the understanding. Okay. At the same time, there was something really weird going on, which said that that simple story that eukaryotes and archaea are the same or more closely related than bacteria was not the case at all. And so this is, this is the spoiler alert, as they say. So eukaryotes are a hybrid of bacteria and archaea. It's like the strangest thing you can imagine. There were two types of cells. You put them together, you get the third type of cell. Yeah. This is called eukaryogenesis. One of the people who really this is not the only person, one of the people who really prominently put forward this hypothesis and built up a scientific consensus around this was Lynn Margulis. And this picture is on her wedding day with Carl Sagan. And when she wrote the paper, she was called Lynn Sagan. So she wrote the first paper on, well, not the first, there were actually some early Russian scientists who had the same idea, but she really found data for it. So she argued that in eukaryotic cells, there are these little objects called mitochondria, which we know very well, right? That these mitochondria actually morphologically, molecularly resemble bacteria. They used to be bacteria. And somehow they ended up inside another cell and became mitochondria and became permanently enslaved. This was called the mitochondrial endosymbiosis hypothesis. And it sounds really vague, and how do you test this? And it turns out there's many predictions you can make from this, and you can test all of them, and it's all true. So this is now absolutely true, that mitochondria used to be free-living bacteria of a type which are called alpha proteobacteria. Another type of alpha proteobacteria is E. coli, for example. So mitochondria are like a cousin of E. coli. And they live inside this other cell. What is this other cell, though? Is that also a bacterium? Right? So I've already told you what it probably is. So mitochondria are descended from free-living bacteria, but what's the host? And now we fast forward, and I would not have had a very good answer to this even 5 or 10 years ago. But if you look at the papers I'm referencing, 2023, this is all hot off the presses stuff. Yeah. So just look at the ages. Prokaryotes, we know, are about 3.5 billion years old. We know this from fossil evidence. Eukaryotes are about 2 billion years old, again, from the similar kind of sources. Here's a very nice paper looking at fossils from, I think, an Australian core. And not only are they doing microfossils, but they're also doing chemistry. You can extract chemicals that are known to be eukaryotic chemicals or prokaryotic chemicals, and you can see when they first appear. And from these kinds of analysis, it's absolutely clear that prokaryotes, bacteria and archaea, far predate the eukaryotes by 1 billion or 1.5 billion years. At some point, the eukaryotes then arise. And so the one is first and one is second. Okay, so this establishes that the root is in that part of the tree. The second thing is now we have, again, evidence from sequencing, but now we have tens of thousands of eukaryotic genomes and bacterial genomes and archaeal genomes. And you do the simplest, the most simple-minded thing. You take a eukaryotic cell, like a human cell, right? It has 20, 25,000 genes. You take every gene and you say, can you find a gene like this outside of eukaryotes? You would expect, if biology worked the way Darwin thought, that they will all be from one group, which is sort of the group that contains the ancestor. Instead, depending on which kinds of genes you take, right? If you take various kinds of enzymatic genes, enzymes, lipid metabolism, ATP synthesis, and so on, those genes from the eukaryotes all map back to bacteria. But if you take genes like the ribosome or DNA replication machinery and so on and so forth, tubulin, those genes in the eukaryotes all map back to archaea. Okay, very mysterious signal. If you saw this, you would think you contaminated your sample. How could it be that the same organism maps back to the two most distant forms of life that you can imagine? Okay, so this is what I mean when I say we, eukaryotes, are a hybrid of bacteria and archaea. It doesn't explain anything, but this is just a fact, right? So how could it happen? We don't exactly know. And now we're in the realm of speculation. So let me give you a sort of picture. Oh, I'll fill that in. Thank you. So this is also meant to be a phylogenetic tree, but not a Darwinian one, because things actually converge instead of diverging. But setting that aside, any tree, if I pick any pair of branches, I can find the time when they were most recently related, after which they continue to be related. So that point of relatedness is called the last common ancestor. Okay, and so what we have here is the last eukaryotic common ancestor, which is like a notional organism, an abstract concept at the root of the eukaryotic tree, the last bacterial common ancestor, the last archaeal common ancestor, and the last universal common ancestor. Now, these are all just abstract notions of any phylogenetic tree. We don't actually know what these creatures would have looked like. So, what's interesting is Darwinian evolution, as we already brought up, is all about this sort of brutish competition, right? It's dog eat dog. There's not enough food. I win or you win. So eukaryogenesis is very different. It's actually a place where the tree comes together and it arises out of cooperation and not competition. So sort of philosophically, it's very interesting. Yeah. Now, it's not clear that being a eukaryote is much better than being a prokaryote, because the biomass on Earth is dominated by both. It's not like one is much more successful than the other. But of course, only eukaryotes have been to the moon. But even that's not true, because each of us take 2 kilograms of prokaryotes inside us. So so it's hard to say. Yeah, exactly. Yeah. So it's good to be a bug. Yeah.
The title of the second paper was actually what when you gave it away.
Yeah.
2 slides ago, I was thinking, is it the difference in what eukaryotes have in terms of what we get from the bacteria comes from the fact that everything in the nucleus comes from that?
No. I'll get to that in the next slide. So what is the difference between prokaryotes and eukaryotes? The problem is there's no one difference. There are so many differences between prokaryotes and eukaryotes that it's what Stenier and Van Niel called an unbridgeable divide. We have a nucleus, and that's the eukaryote means there's a true nucleus, right? But we also have a cytoskeleton. We also have internal membrane bounded compartments. We also have mitochondria. We also have sexual reproduction. We have mitosis. We have the spindle. So there's too many things. Now, the genes that are responsible for all this, almost all of them are now in the nucleus. Okay, some of those genes used to be in the mitochondria, but over time there's a process by which the mitochondrially localized genes, mitochondria also have their own DNA. Okay, but there was a time when all the mitochondrial genes were in the mitochondria, but there's a process by which those genes end up going to the nucleus by fragmenting the DNA and going inside and being fixed in there. So knowing where the genes are don't tell you where they came from. Okay, thank you for the question. Okay, so what's interesting about eukaryotes? It's their internal structure. That's it. Okay, and you can see when eukaryotes first arise in the fossil record by just documenting the kinds of internal structure being very modern in nature. So modern looking eukaryotes, I mean, I think the scale bars are like 10 or 20 microns. Sorry, I should have written it down. But eukaryote looking organisms end up in the fossil record. I met a sort of eukaryotic fossil hunter, Emmanuel Jabo. These are microfossils. Okay, what she does is she digs through rocks, and most of the time there's nothing. And every once in a while you find a slice where there's a cell fossil. Cells are very soft and they don't tend to leave fossils. And yet we have things like this, right? So eukaryotes arise in the fossil record with internal architecture about 1.6 billion years ago. And so here's the picture. Here's the cartoon, right? If you want to just hang your head on a hypothesis about how all this crazy data I'm trying to explain works, this is what we know. We now know that mitochondria were like alpha proteobacteria, and that the host cell was an archaeon. So there indeed was an archaeon, a singular of archaea, and a bacterium that somehow came together. In fact, we know the closest cousin of that archeon. The reason we know this is there was a deep sea expedition off the coast of Norway where they dredged up these things, and these cells don't survive when you bring them up to atmospheric pressure. They just explode. But you can get the DNA and you can sequence it. And this organism, which was called Loki archaeota because the deep sea vent is called Loki's castle, had genes that previously were only found in eukaryotes, is now found in an archeon. And nobody knew what it looked like. And it took 10 years for somebody to culture it. And in the end, it was cultured in 2020. And there it is. It is a very morphologically interesting bug. Here's what E. coli looks like. And somehow these two things, which, small and not very interesting internally, come together to make, a very complicated cell like this. How does that happen? So here's the cartoon. And you may ask, by the way, after all this time, where's the physics in this talk? And I'm telling you, it's coming up. And what I'm trying to argue is that the origin of eukaryotes, apart from all the biological questions you have to explain, there's also a physics question you have to explain, okay? Which is that somehow you had these cells with very little internal architecture apart from just having DNA, collapsed in the middle, combined together to make this kind of cell. And the current understanding, which is fairly well grounded, is the following. You had these archaea with these long tendrils, these so-called processes, and those tendrils increase the surface area of the membrane. In those tendrils, you know what happens? Bacteria actually sit, and this is true today. You can find cells that have this kind of external symbiosis. Over the course of time, if that external symbiosis then becomes deeper and deeper and tighter, this is like a long period of dating, before you finally decide to get married, and it becomes irreversible. The mitochondria then, just by the membrane expanding continuously, appears to become engulfed. Actually, topologically, these two are identical. And then there's a topological change, which creates the outer membrane, and what used to be this cell remains in a remnant as the nucleus. Okay, but it still doesn't explain where all the other membrane architecture of the cell comes from, which is absolutely stereotypic for eukaryotes. And that membrane architecture arises dynamically and physically through processes we understand microscopically, and this is what we study in the lab. Okay, so our interest is the origin of eukaryotic organelles, which are these internal membrane-bounded compartments, like the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and so on and so forth, and the cargo, the traffic that moves stuff between them. Just to say again, to recapitulate this, there was an archaeal bacterial symbiosis, which became a mitochondrial endosymbiosis with the simultaneous origin of the nucleus. Moderate data to support this. But after that, there was a period of rapid diversification of eukaryotes. Eukaryotes became more complex, and like anything at the base of a radiation, you get a huge variation of form when you enter a new niche. We don't know what the original niche that eukaryotes became very good at surviving in, but there usually is a big radiation. The problem is that you can imagine time going to the right, from proto-eukaryotes. This is the LICA again, last eukaryotic common ancestor. As you go over time, there's this tree. And if you were so lucky that extinction never happened, then you'd be able to retrace the order in which all these different eukaryotic features arose along that billion-year branch. Nucleus came first, then came cytoskeleton, then came sexual reproduction, or whatever it is, right? Because you would find intermediate forms. How do we know about human evolution? Because the fossils give us intermediate forms. For example, hominids that were not always fully standing, or the cranial size changes over time, right? But cells hardly leave fossils. So once everything has gone extinct, all you're left with is the long branch that started here and ended up uniquely at the last eukaryotic common ancestor, because all the intermediate forms which could have survived till today died out. And unfortunately, or fortunately, depending on how you look at it, last eukaryotic common ancestor, based on all the genomic reconstruction we can do, was 1.5 billion years old, let's say, and as complicated as any eukaryotic cell today. Any complex thing today's eukaryotic cell had, that cell had. Okay, so all the complexity arose along this branch. So how do you study that if you're not left with any samples? And I would argue the only way to study that is to model and simulate this process and see what we can see based on our understanding of the underlying mechanisms. After this, of course, there's huge diversification of eukaryotic lineages leading to plants, animals, and amoeba, and unicellular ciliates, and various kinds of parasites, right? All that good stuff happens. But surprisingly, the cell biology of eukaryotes doesn't change over time. This is why in biology, when you get into biology like I did from outside, everybody tells you, read this one book, and it's called The Cell. And it's not called, this cell or that cell. It's just called the cell because cells are so similar internally that you just have to read one textbook about them, no matter which organism you're working on, except for bacteria, which nobody cares about. So that's a joke. Okay, so along this, so then over the years, I've had the privilege of working on different pieces of this hypothetical history. We've actually worked on the archaeal origins of eukaryotes by looking at the sequence of Loci archaeota. We've looked at the mitochondrial integration and discovered that there are still some intermediate forms for that. For example, the seaweed from which we get this agar-like stuff that goes into ice cream and toothpaste, and you find it on the beaches, this slightly red-colored seaweed, retains a very ancestral type of mitochondria. We've looked at the origins of exocytosis and endocytosis, and eventually the origins of very complicated organelles. My favorite organelle, which is called the Golgi apparatus, which I'm not going to talk about today. But the point is, so there's still a lot to fill in along this very long branch. Okay, quick snapshots of what we did. So we actually looked at, along with Busbaum and Gautam Day, the genome of Loci archaeota compared to the genome of eukaryotes, and found out, again, which is the origin of various unique eukaryote type proteins. And you can see the Loci archaeota contains at least some interesting proteins that could not have been ascribed to a bacterial ancestry, like these small GTPases, which if you're a biologist, you'll like them because they are responsible for membrane labeling. This is the ancestral mitochondrion. We reconstructed using deep time phylogenetics over 1.5 billion years and found that 3 eukaryotic groups today contain these ancestral type mitochondria. For some reason, those mitochondria haven't changed over time, and that's why you might call them living fossils. And more recently, I started getting interested in how eukaryotes can generate the underlying genetic machinery to change their cell biology, right? There's a common theme in biology that evolution requires a dynamic substrate, just point mutations. Point mutations are not enough because typically a point mutation will kill you. It's not going to lead to any huge innovation. And so there's a common theme in biology, which Ono first identified, which is that duplicate gene copies, which arise by chance sometimes when the DNA replication machinery stutters, it makes a second copy of a gene. When you have 2 copies of a gene, well, then by accident, if you break the symmetry, one of them gets locked into the ancestral function, the other one can evolve a new function without breaking what was already there, which is very important. So gene duplications are very well known to drive eukaryotic diversity. So there's one mechanism of gene duplication which is quite common in nature, where 2 different species hybridize. Again, the textbooks tell you this doesn't happen. It does happen. And when they hybridize, you have 2 copies of every gene. And now the world is your oyster, right? You can make all kinds of changes. But typically these 2 copies don't do anything for you. They are the same. You might as well just have one because you energy to keep the extra copy. So you lose the extra copy. So it turns out in the ancestry of yeast, which is the cell that makes bread and beer and so on, there was a whole genome duplication 100 million years ago. So we looked at the cells that survive today from the whole genome duplication, a very broad spectrum of yeast that survive today. And the ancestral cell had 5,000 genes each. They hybridized, you made a cell. Yes?
There are several instances of entire genomes.
No, this is how they duplicate. Whole genome duplication typically happens after hybridization, not spontaneously. But I can tell you technically why that's the case. Okay, because if it happens that way, then you don't produce viable sporulating cells. Okay, so anyway, so the ancestral post-hybridization cell had 10,000 genes, but these are expensive to keep. You don't want to keep them, so it loses it. And so if you look at present-day cells, they all have 5,000, not 5,000. They have about 6,000, 5,500. So they kept about 10% of their extra gene copies in double copy. And if you look at what the function of those genes is, 2 become enriched. So one is ribosome, which I haven't shown, and the second is vesicle-mediated transport. So for some reason, extra gene copies seem to be implicated in the system I'm talking about, which is vesicle trafficking. It seems to be a good substrate to drive the evolution of that. So this got us interested in modeling this whole process. Oh, no, it's it's 8 or 16. Or 32, it's powers of 2 because you keep hybridizing. Okay, yeah. So you know exactly how many hybridizations it's been if you look at the powers of 2. Okay, it's very cute. This was not from some time ago, but so again haploid diploid also. So 16 chromosomes is your standard use. Okay, What is vesicle traffic in the short time that I have left? And just remind me, how much time do I have? Good. I'll finish. Now we're into vesicle traffic. So this is really going to be the heart of my talk. All this was just a prelude to why we work on the stuff we're working on. So what is vesicle traffic? It is the basis for this complicated intracellular organization of a eukaryotic cell. This is meant to be a slice of a big spherical eukaryotic cell. That's the nucleus on that side. This is the plasma membrane here. This is the rest of the universe over here. Inside the cell, you have these internal membranes which contain within them some other space where complicated stuff happens. That's the dark gray. Okay, so you have the ER, you have the Golgi, you have the lysosome, the endosomes, other kind of granules. This thing, if you look at an EM image, it looks like, okay, it looks complicated but tractable, right? Because you can imagine putting all these things in, just like I put mitochondria, you could put a Golgi or you could put a nucleus. But that's not the way it works, because every one of these membranes is rapidly exchanging material with every other membrane in the cell. And what it's losing is not the same as what it's gaining. Okay, so the very fact that you seem to see a fairly constant so-called homeostatic cell organization, despite this major exchange of material underneath, shows that it's some sort of a tractor state, right? It's not spontaneous organization, it's highly controlled to be this way. And all the control is happening at the level of proteins and their interactions. Okay, so that's just remember that you have a bunch of different compartments and they're exchanging matter left, right, and center. So why don't they change? That's the question. So there are two types of emergence we've been looking at, right? The emergence of evolutionary time, which I've already mentioned, but also this second type of dynamical emergence, the emergence of this structure from elementary molecular processes. Okay, it arises, it's an emergent structure. It's a classic example of emergence. Now, zoom in again. Interestingly, after all these years of doing cell biology and several Nobel Prizes just to explain this slide, okay, we know how vesicle transport works, and a lot of people here study exactly this process. So you know, we're in one of these places where if you wanted to have experts working at every one of these steps, you will find them. Yeah. So we just had great discussion about snare-mediated fusion, right? For example. So let me just say everything in summary. You have a donor compartment. You have an acceptor compartment here, also called a source and a target compartment. There's a cargo inside each one of these compartments there. And that cargo, that yellow cargo, has to be delivered over here. So you can think of warehouses, and there are trucks moving stuff between warehouses. If it was Amazon's logistics system, it's easy. The truck comes, it says, here's what I need to collect. You give the driver the stuff, he goes, he sees, here's where I need to drop it, he drops it off, okay? In this case, the thing that chooses the cargo, they're called coat adapter proteins. They're the ones that basically make these budding vesicles happen, and they reach inside through transmembrane domains, and they pick up the right cargo and the right transmembrane cargo. So this vesicle, by the time it's made, has three types of cargo: that purple guy, the yellow guy, and the red guy. Okay, so choice of cargo is very specific. You don't take all the cargo that's there. Secondly, unlike Amazon, where the cargo is one thing, but the manifest that tells the driver where to go is written on paper or on his iPad or whatever. In this case, the cargo itself tells the truck where to go. Okay, so it's a self-referential nature to this, which makes it mathematically exceedingly complicated to understand. This is the heart of what makes this a complex mathematical problem. Because in this example, that red guy is called a V snare, a vesicle snare. It's a type of protein which actually matches in a lock and key fashion through specific protein-protein interactions with this triple helix bundle, which is called the T snare, and delivers the cargo via fusion of the vesicle. In the cell, there are dozens of different types of V snares and dozens of different types of T snares and dozens or half a dozen type of adapters and so on and so forth. These molecules, which are homologous, means they came ancestrally from the same protein, but there are many gene copies. Okay, so this is interesting. So let me just summarize here. What are the two main mathematical problems? One is that for homeostasis of the compartment, whatever goes out must match whatever comes in through all the vesicles that come in and go out of a given compartment. Secondly, somehow the machinery that regulates budding is the same machinery, but different type or copy in different parts of the cell. And the machinery that generates fusion is, again, the same type of machinery, but different copies in different parts of the cell. So there's been always this sort of vague hypothesis in cell biology that the duplication of these genes at the genetic level literally leads to the duplication of compartments in this very one-to-one way. And that I've always found pretty mysterious, how you could make that claim, right? So we wanted to test this claim. So to test this claim, then we need a model, okay? I can't test it in real life. It's still It may not be later, but it's still too complicated to recapitulate all this in the tube. So we wanted to make a single model, which I'm going to outline for you today, which contained all these different layers of interest to us. Okay, there's genes, which can duplicate or delete or diverge. There's modules, which are these interacting sets of proteins that drive budding and fusion and regulate the processes. There are networks, which are these flows inside the cell through different compartments. And finally, in any biological question, there's function, and therefore there's selection in the Darwinian sense. So we wanted to make a model. I mean, it seems like a big ask. It is. That contains all these pieces so that we can finally understand by selecting on top, how does it influence the genes on the bottom and the networks to mediate it. And it's not that this is new. We and several others have been doing this for many, many, many years. We use different methods. I'm just flashing this here to show you that you can make sort of a dynamical systems model where the composition space is very big and you find dynamical attractors and the basins or sometimes chaos. You can make these Boolean models, which are much more rapid to study, and they lead to, these kinds of models lead to very simple topologies. These Boolean models lead to rather complicated topologies, but they don't contain any notion of genes or gene duplication. Most recently, quite excitingly, we've managed to combine results from all these models into a sort of graph theoretic framework where, in fact, some theorems from the 1970s about how literal traffic moves around cities and how you should regulate traffic seems to come back here to explain how many different kinds of branches a cell traffic system must have. So this was the past. So today I'm going to tell you about another model, and it's very new. This is what I'm going to talk about. It's abstract. It's so abstract and simple that you can take a piece of paper and you can carry out all these calculations yourself. There are no calculations. They're like puzzles. It's like doing Sudoku. Okay. So here's the model. Remember I said there were these coats and adapters that make the vesicle? There are many different types. So imagine a cell that has three types: coat A1 or adapter A1, A2, and A3. And I'm just labeling them red, green, and blue. Okay. Any cargo in the cell that moves along these trucks, these vesicles, has some preference for these three kinds of coats. It'll either like none of them, which is the 000, or it'll like all of them, or maybe it only likes the red and the green, or, whatever it is, right? But in principle, then with three coats, there can only be 2 to the 3, 8 types of cargo. There can be thousands of types of proteins, but they fall into these 8 classes. And in the obvious way, then you can color code the cargo in the usual way, like red plus green is yellow. So you get these 8 types. The black type doesn't like to go on any vesicle. The white type likes to go everywhere. Everything else has some sort of nuanced preference. The objects of the model are compartments, which are these big guys, which contain cargo, and vesicles, which are these small guys, which are color-coded by their coat on the outside and their cargo on the inside. So, for example, the green vesicle is a green-coated vesicle. It can only contain yellow, green, and cyan cargo. It can't contain red cargo because green vesicles don't like red cargo. But you can just look at it here. It doesn't pick up red at all. And of course, it won't pick up that cargo unless it's in the compartment. So you would think you can't get very far with this kind of abstraction, okay? But it turns out you can. So there's compartments, there's vesicles, and there's cargo. Then what do? You walk over to your colleague who's studying some type of cell and you ask her to draw what she thinks is the topology of stuff happening in the cell. And this is the kind of diagram you typically get in a textbook. It's a very highly simplified representation of stuff moving around. I've already showed you 2 or 3 examples of this. So the ER turns out, and I won't belabor this, but the ER is important and different from all the other compartments because that's where you synthesize lipids and proteins. So everything is made over there, moved, and potentially degraded elsewhere. So that's okay. So a hypothesis for what a cell looks like, you have to say what is the ER, and then you have to say which coat actually moves cargo on which vesicles. Okay, this is just my guess of what a cell looks like. As soon as you tell this to me, then using the rules of what cargo likes to jump on which truck or bus and move around the cell, you can immediately, in a unique way, figure out what the compositions of all the compartments are. Because after all, everything is made in the ER and it can only go to some place. So eventually it will be in some collection of places. Okay, so this is unique. If you give me the hypothesis of the network, I'll tell you exactly the compositions. Unfortunately, this is not the whole story because these compositions have to be explained in terms of underlying molecular interactions. So remember I told you that budding was regulated and fusion was regulated. Okay, so let me just explain to you what that means. It means I have to posit some sort of functional molecules, which are, for example, here are coats. I have to have a recruiter for a coat that drives budding. Here's a V snare, here's a T snare. This is an activator of a V snare that regulates it. And I have to have all 3 at the right place at the right time to drive fusion. Here's a specific example. If I have this red vesicle budding from this compartment, you should say, why does it do it? And you could say, well, maybe the red coat likes the cyan label. In which case, you should also have a red code coming out of this compartment, and look, you do. So this is a consistent guess. Similarly, for fusion, I can say that the magenta and the yellow snares like the red snare, and they should fuse, right? So this seems, a lot of things you have to take care of, and there is, and you have to write down all the things you need to do and all the things you need not to do. And eventually, for a network like this, I can come up with a molecular hypothesis for how it works. Okay, and if you just take a photo of this and go home and work it out, you'll find that this is exactly a solution to that problem. Okay, how does the evolution happen after that? So I know I'm running through things a bit, but remember what I have now. I have a topology or a network, then I have the molecules that make the network. Now I have to let the molecules evolve. What I'm going to say is the molecules evolve by gaining and losing their interactions with the coats. Remember, it's a 3-coat system, and so it basically involves things changing colors along this little cube. So for example, I can go from this molecular triplet to this molecular triplet by 2 steps of mutation. So this is the underlying evolutionary space. Okay, so now what did I do? Somebody told me, here's the network. Then by working very hard, you said, well, here's one possible solution. Well, it turns out that you can write down the conditions to make this network work as a sort of Boolean satisfiability problem, which is a standard way to write down expressions in computer science. And the Boolean satisfiability problem says which molecules you need or don't care to make something work. Okay. Just to flash the numbers, in this particular simple model, there are 3 kinds of coats. There are 2 to the 3 recruiters. There's 24. There's 2 to the 3 possible ones of each of these 3 rows. There's 512. So the possible space of answers to the satisfiability problem has 536, 512 plus 24, zeros or ones. Okay, and the solution space, because the satisfiability problem says you can't have any ones in this red zone out of 536, and you must have at least 1 1 in these blue zones. That's how the solution space works. And once you have this, it's very easy to see what's going on. Now we can do evolution, because for any network you draw, I can tell you what the solution space looks like, and so I know how the solutions connect to each other by mutations. And just by sampling, we find all of the following things. So this is the space of all possible solutions. It's 2 to the 536. Okay, and the different kinds of networks. This red, green, blue has nothing to do with the other red, green, blue. Sorry. Different types of networks. For example, the blue network, the red network, and the green network. What this means is, in this part of the solution space of molecules, the network that you get is the green type of network, which I haven't shown, but it's some particular topology. So, for example, you can have one network with 2 different families of solutions. Or you can have, so the same network can happen through different families. Or you can have 2 different networks with the same solution that works for both of them, even though the expression is different. The same Boolean variable can satisfy 2 completely different kinds of clauses, right? So that's a place where it's bistable. Around there, the same cell could make both networks depending on how it's nudged by initial conditions. Or, this is even more interesting, you can have a kissing region where just one shift can make the old network go and the new network come. Okay, these are very interesting things. But mostly, do you know what you get? You get impossibility. You get a condition where something that has to happen is inside the region where something has to not happen. It's a contradiction in the Boolean expression. The system just doesn't work. Whatever hypothesis you had is not satisfying. And that's the largest part of the space. Okay, so what we did was we sampled billions of network topologies, and we just ran through this exercise and said how many of them work, how many of them don't. For small networks with 1 compartment, 2 compartments, and certain number of edges, you try a few thousand networks, you get a few thousand solutions. I'll explain what the different columns are in just a second. But once you get to large networks, you can test billions of networks and get like 100 or a few hundred solutions. Okay, so and this is very common in sort of Boolean satisfiability. You make the system big, something is going to spoil the answer. Okay, so these systems are highly non-random samples of the molecular space, which any biologist would know, but it's nice to get a quantification. These different columns, I'll just flash here. There's actually a very interesting question in cell biology. When a vesicle delivers that V snare, it later has to recycle that V snare. And when the V snare comes out of the source compartment, why doesn't it just fuse back? Because it contains the same label. So snares must be regulated in some way. We still don't know exactly how. So as a way to get that biological detail in, we tested many models by which the snares were regulated. So unregulated, activator, inhibitor, and so on. It's not very important. The important thing about the slide is you take billions and you get hundreds. Most of the space is just not viable. Okay, finally, the last thing I want to talk about is function. Now I have this network, right? Or I have this network here. How do I talk about function? I don't know, because I don't know what these cells want to do. I don't know if they want to do phagocytosis and eat. I don't know if they want to secrete some factor which influences the environment. I don't know what they want to do. So we were stuck on this point for a long time until we had the idea, let's not select for function, let's select for information. A cell that is more information rich has the capacity to carry out many more kinds of function. Okay, and so let me explain in detail what I mean, because this is quite important. So in design theory, there's something called an affordance. An affordance is a feature of an object that makes it self-evident how to use that object. So for example, the color of the cap seems to indicate it's different from here, and you would think you should twist the cap to open it. So the most classic example is on a door. If you have a flat piece on the door, you should push it, and if you have a handle, you should pull it. So it's an affordance that makes it obvious to the user how to use the object without any detailed underlying theory. So we said, let's look for affordances in these vesicle traffic systems. So for example, here's a network on the left side where the red label is only on this compartment and the black label is only on that compartment. So any function you want that would like to separate itself into two compartments, if it learns to live with the red or live with the black, it's very easily able to do that. Okay. So this uniquely being able to, so think of a city with bus stops and buses moving between bus stops. If you're able to just look up where you are and very quickly realize where you are in this bus stop network, that's really useful to you to be able to navigate the city and do many other things like go do your job. Secondly, also, it's not just the bus stop, but also the buses. It'll be nice, and it's not always true in Bangalore, for example, if you get on the same bus at the same bus stop, it goes to the same place. Yeah, so a bus that is reliable and regular in that way will then become a substrate for certain commuters to rely on over time, right? So these are all substrates for function. So here's a network where the red label, the black label, and the magenta and the yellow arrows are all these information-rich labels. Here's another network, which turns out to be reachable molecularly from the first network, which has, it looks more complicated, it has exactly the same function. Because it has the red label, it has a black label, it has exactly the same transfers. Okay, so this is, looks more complicated, but functionally is the same as that one. Evolution doesn't care, you could go back and forth between them. Here's a better example. So here by one mutation, I can take this network to that one. And what happens? The original function is preserved, there it is, but a new function emerges, a new place which is labeled. Over time, new functions will sort of glom onto this blue label. And once you get used to that lifestyle, you can never go back. And I don't know how long that time is, but there's some sort of time to stabilize that new function. So generally, the movement from here to here will be asymmetric, unlike the previous case. So this is the ratchet I need to increase the complexity of the system. So I'm going to bring it all together now. So what do I have? I have the space of all possible networks, practically the space of all possible networks up to a certain size, because I'm running out of computational resources. Okay, so we've gone up to networks with 4 compartments, but there are billions of them, out of which tens of thousands actually work. Okay, so there's 21,000, for example, labeled networks with transitions. Okay, each network you can imagine as a dot in some large space. Then I have these transitions, which can be unidirectional or bidirectional. So that's a graph in the mathematical sense. So in this graph, there are all these islands, which are large collections of networks linked by unidirectional changes. I can flow across all these back and forth, back and forth, back and forth, changing what's the genes and everything else, but the function doesn't change. It's the same. So this is what biologists call a neutral space or a neutral walk. It functionally doesn't change, although in detail things change. You can also think of this as a large number of microstates that correspond to the same macrostate in some sort of statistical physics model, because the macrostate is defined by very few features. The microstates are highly detailed. And so you can do statistics on these islands. And you can say what's common to all these things. Then you have the unidirectional reactions, which connect one island to another, only going one way, because you gain function. Okay, so I can place these tens of thousands of networks into all these little islands, which are neutral internally, but functional externally. And then I'm just going to compress this very large graph of 20,000 nodes into this structure here. Inside each of these little gray boxes are hundreds or thousands of individual graphs, but on the x-axis is the number of labeled bus stops, and on the y-axis is the number of labeled buses. Okay, and so going to the right or upwards or both is functionally beneficial and typically irreversible. And each of these little sort of motifs inside there, all the networks have exactly that function. For example, here you have 2 compartments and 1 edge. Here you have 3 compartments and 2 edges. Now you can imagine that having 3 compartments and 2 edges, you can write this down. There's many different ways to do that topologically, right? It turns out that not all of them show up. So here you see the sign of not selection, just basic physical constraints. You cannot make a network with a certain functional motif using the kinds of rules we know that snares and courts behave. It's just not possible. They don't arise, right? So for example, within this island, there's only 4 motifs, whereas there should be like 7. Okay, so there are missing motifs, which is a sign of just basic physical constraints. Secondly, there's what's called contingency in biology, which is the history dependence of biology. Where you are constrains where you can go. So for example, from the bottom here, you can actually go many different directions, but from this region, it looks like there's very few ways to get out. And in fact, I'll show you in the next page, there aren't any at all. So you can look at the flows in this network, and these are evolutionary flows over time. And remember, just lest you forget, I'm talking about now the flow of the internal complexity of the eukaryotic cell over billions of years, dominated by long neutral walks inside these islands, and then occasionally a jump to a higher state of complexity. And you can model this, okay? Questions?
What determines the size of this item? Is there a critical size that determines whether the network is more evolvable or not?
No, so we have all the data, and now we have to look for these correlations inside the data, and that's we've been sifting through to look for patterns endlessly. So this is where we are at now. I've reached the point where I think we'll give it to some machine which can give the answer for me overnight instead of But we found some structures, and yes. So for example, there are networks that are quite fragile, and many things you do will break them, and so you have very few ways to get out. Yeah, is that the kind of thing you're talking about? Yeah. So for example, so these 4 are exactly the same model, but for different types of assumptions about snares and the regulation. So here there are 2 coats and a snare activator, 3 coats, a snare inhibitor, 3 coats, not regulated snare, 3 coats, snare activator. So you can see, for example, going from 2 coats to 3, you vastly increase the number of possible topologies. And secondly, you see that going from an unregulated state to a regulated state, you vastly increase the number of topologies. Again, intuitively obvious, but it's very nice to see it happening in a model. Also, very interestingly, the contingency of evolutionary history is stark over here. If you end up in that system, which is 2 compartments with bidirectional interactions, you're stuck there forever, at least with this number of coats, and you can't get out. But what's nice is that if you start off from a proto-eukaryotic cell, typically a large number of trajectories, I mean, it's not overwhelming, but let's say a majority of trajectories end up with 1 type of topology uniquely selected. So this is the type of convergent evolution that happens. And therefore, when you're looking at this evolution of proto to complex eukaryote, you can ask, is this thing sort of looking, smelling a little bit like a modern eukaryote or not? And so you can go inside in great detail and you can see what kind of networks you get. And interestingly, a lot of things that cell biologists care about, which is, for example, taking proteins from the ER through the Golgi and secreting it, or doing endocytosis and recycling of receptors, or doing phagocytosis and eating food, all those things start to happen only when you get up to that level and not before. Okay, so it could have been that there was no way to get to that level, but there is. It could have been that way is very rare, but it isn't. It could be that there are ways to get out from that level, and you need more underlying types of molecules to get out. I'm not saying that's the end of it, but in this system, if you try and get out from there, what happens with only 3 coats is you break something else. So this is a very common theme in biology. Once you make something very complicated, you're scared to touch it. I mean, this is, for example, why Microsoft's code in Windows is absolutely horrible, because all this old stuff they can't touch. Everything depends on it. Okay, and so therefore you have to work at some other level. In biology, it responds by going to another level of selection. When the genetic code got too complicated to tweak, it went into sort of chromosomal architecture and gene regulation. When a single cell became too complicated to work with, remember I told you these eukaryotes haven't changed in a long time, right? They changed from proto-eukaryotes to these guys, and then they froze. Then where did the innovation in biology come from? It came from multicellularity. And then it came from social organization, like bees and ants and humans. Right? So at every stage, when something gets too complicated to stop functioning, it's a common theme in biology. You go to another level. This model contains that as an emergent property. Okay, so that's it. Here are the lessons about from this model. To be fair, and none of this is a statement about biology. It's a statement about a model that's rich enough to describe some simple biological systems and doesn't make any obviously wrong assumptions. There are evolutionarily plausible routes towards intercellular compartments and complex traffic, which is interesting. Molecular constraints lead to constraints on the topologies, which is interesting. However, there's no one-to-one code where I can see the molecules and get the compartmental architecture. There's a huge amount of degeneracy or redundancy in terms of what solutions one could use to make a particular architecture, which is perhaps why the genetics of vesicle traffic has been so difficult to crack, right? There's too many different ways to make something work. The trajectories themselves are not the simple idea you duplicate a gene, you get a new compartment. They're dominated by ultra long random walks, and you can make this precise. You can ask how long it takes to escape a Markov process from this big island, right? And it takes exponentially long. Luckily, you've had billions of years, and each step is like 1 day. So maybe it's okay, right? Maybe that's why it took so long. And there's no simple link between new genes and new compartments, which is what I say. So I'm going to end. This is a slide from related to a paper that came out in Nature, which has been controversial, but I find the ideas in it interesting. It's called assembly theory. And assembly theory says, how did complexity arise over time? It's a sort of theory for the origin of life. I'm going to use some of the words from that theory, we don't use the theory itself. There's a universe of possible graphs, which is a purely mathematical concept. Out of those graphs, there are things that are molecularly possible by the physical interactions and regulatory interactions needed to make that graph. Out of those, there are those that are reachable in evolutionary steps over a reasonable amount of time. Those are called contingent. And each is a subset of the previous one. And out of those, there's what's actually observed. And each one of these filters decreases the set of possibilities you want with the hope of getting a small number of tractable possibilities at the end that you'd like to study. So I'll stop over there. Here's the group, and the work I've shown you is mainly the work of Sahana, who's a PhD student at NCBS, and Kritika, who's a master's student from IISER Pune. Everybody else works on other things in the group. You can read about it here. And I was speaking this morning to Deshmukh students about how I got here, how we chose this problem. We had a meeting at this place called the Kavli Institute for Theoretical Physics in Santa Barbara 10 years ago, when I was at a sort of despondent phase in my life when none of my experiments worked, and I wanted to look for a new problem which combined evolution and cells. So I thought the best way to do it is to call a bunch of cell biologists, evolutionary biologists for 3 months and just talk to them. And I made notes from this meeting, and everything we worked on from that time, including all this stuff, basically arose from that sort of interaction. So I guess the lesson is, at some point you need to break out of what you're working on, regroup, and come back to it. It's always beneficial. So thank you for your question and attention. I'm happy to take more questions.
More questions?
We've had quite a few already. In the back.
I was wondering, since you mentioned that your networks evolve,
Yeah.
do you have networks that never stop evolving?
Yeah, so these networks never stop evolving, but they end up moving in cycles in large spaces which are neutral. And this is a very common feature also that you see. It's not that we have stopped evolving, okay? We keep moving neutrally and don't gain much function. That happens at the level of a single species. In biology, when you have an arms race between two species, those networks also never stop evolving, even though the function may not improve dramatically. You make a change, they make a change, you make a change, they make a change. This is called the red queen dynamics, okay? So most things at the micro scale never stop evolving. Yeah. You seem disappointed with that answer, but okay. Yeah.
So if they never stop evolving, like what is the characteristic of that
Very good.
network?
So I know one which very clearly has stopped evolving. It's the genetic code. It's the dictionary between triplets of nucleotides and which amino acid it encodes. Every type of cell on the planet, except for one, which is ciliates, have the same genetic code. There was some point when it stopped evolving. What is its characteristic? As it happens for the genetic code, its characteristic is optimality. It's optimal for something, but that's not guaranteed. So Sidney Brenner said that biology is the art of the good enough. I think something like that he said, right? As long as something is good enough, it'll get stuck. You don't have to get optimal. You just have to be better than, what's that joke? You don't have to run faster than the bear. You have to run faster than the next guy, right? So it's like that.
Okay. So what decides, which cargo for which coat?
Uh-huh.
And can you fool the system and make it take a different cargo or a different
Yes.
boat?
So we actually collaborate with, for example, Alberto Luini in Naples. There are transmembrane cargo that have these very small amino acid tails that stretch out into the cytoplasm. It's 4, 5, 6, 7 amino acids. And if you look, there are motifs in those amino acids which are specific to certain coat adapter complexes. Okay, so it's just that. So it's just like you think of bacterial transcription regulation, transcription factor, small motif. It's like that. You can swap the tails, it goes to the wrong place. It's just like you think. Yeah, it's very simple.
Okay, I'm not quite sure how to phrase this question. A little bit wandering in.
Yeah.
Okay, all of your, the second part of your talk was about processes, sources and sinks within a cell, theoretical cell, but a cell. You began with the very gripping point that all life on Earth is, its building blocks is a cell, and that it didn't have to be so.
Yeah.
Is there a theoretical planet in which there is a diversity of building blocks, potentially exchanging material? I mean, what you're saying within a cell between, I mean, what does that look like, and would competition necessarily collapse everything to one type of building block at the end of the day?
Very good. So you asked your question, you answered it, but I'll just expand it again and answer it for everybody else. Actually, it's a very interesting question you've asked, right? So if I were to make autocatalytic sets at the origin of life and see what comes out, it doesn't have to be you get one autocatalytic set. You could have many with very weak couplings between them. Either then these become integrated or they don't. And this is the question of selection incentives to move to the next level of selection. Okay, the question is, can you have cheating, and can you be a freeloader, and can you then grow disproportionately to the other one and get extinct? The amazing thing about eukaryotic cells, metazoans, is that we've convinced all the cells in our body to hew towards one goal, which is my fitness and not their own fitness. Of course, there's exceptions, which is cancer and so on. So I would expect that these sort of drastically distinct populations using different building blocks but sort of eating each other at a very coarse level would be unstable to long-term selection, but doesn't mean that they couldn't exist for eons. So it's possible to all of them, right? So it's entirely possible that you get very different forms of life coexisting on the same planet. Okay, but there's one funny bit to life on Earth, and so this is the what is it called? The economies of scale. Okay, why is it that the genetic code has not changed over billions of years? The genetic code is like a human language. Human language travels with human populations, and over time, you just need to talk amongst yourselves. You don't need to maintain your conversation with the ancestral population. Okay, so just by diffusion, you will change the language over time to the point where they become mutually unintelligible. There's no reason for languages to stay the same unless you have some reason that you want to communicate across the entire large population. On Earth, that force is well known. It comes back to viruses. Viruses are agents of massive genetic exchange. They take stuff from one species, put it in another species all the time. And these are very useful bits of genetic material, which if I can just take, it's like an app. I can just run the app, then I have a new function, right? So there is a role for this commonality of language and commonality of function. So if you have two innovating ecosystems, the bigger one tends to be more rapidly innovating, and that's an unstable dynamics, and it'll get wiped out. This is all just very abstract and theoretical. The kinetics of that may cause these states to be prolonged for a very long time. So, I don't know. But it's a wonderful question.
Yeah. Hi, so I'm not from biology background, but I just want to know, like, if you are going from a common ancestor to an quote unquote ideal cell, how do you decide that there can be one pathway or multiple pathways of evolution? And if there are multiple pathways, then what is the most optimal one, or how does biology work?
Yeah. So first of all, I think a lot of evidence suggests there's always multiple pathways. Okay. And when you say ideal, you mean, for example, functionally optimal for this environment, right? Yeah. So there's always multiple pathways. And what determines which one happens is highly stochastic. It depends which decisions you made early, which then restrict your choices for what decisions you can make later. And I think Rich Lenski's experiments in bacteria have really shown this in a lab-based experiment. So this question, you would think it's difficult to answer, but he's been evolving E. coli for, I think, 40 years now. And he's
Okay.
found pathways for a certain kind of optimal expansion.
So do you see like same kind of E. coli coming from two different evolutionary pathways?
You can see the same outcome from two different pathways. Yes, you do see that quite a lot.
Oh,
And you also see the other thing, which is that one pathway is chosen because of the past.
oh,
You see both.
thank you.
Okay, if you guys have no more question, then we stop here and we thank Mukund again.
Thank you.
Fantastic talk. Yeah, please enjoy snacks outside the West Wing.